Dear friend, Namaskar. Welcome to my YouTube channel DP Forensic Lab. Today video I will discuss about IR spectra analysis for forensic use because we know for the identification of any compound, material or the comparison of the two samples, it is very essential to compare the peaks observed in the IR spectra. So whenever we are going to compare the peak, we have to know the detail of the peaks. The peaks in IR spectra is coming due to the which it is very important. Suppose in case of if we don't know about the detail of the peaks by which these are coming, we can interpret wrongly. As suppose in case of the environmental, due to the water and the carbon dioxide is present in the atmosphere. So sometimes these peaks come in the IR spectra, and only seeing that these peaks are matching, or we say this material is same, this may be wrong. So it is a very important to know about some detail about the functional group or IR spectra peaks. So this can very useful for comparison or identification of the material for the forensic purpose. So the whole spectra, this is a MIR spectra from 4000 to about 600 centimeter inverse. This is a spectra, IR spectra, MIR range. So this whole spectra are divided into two main groups. One is a function group region, or this is a fingerprint region. In this fingerprint region, there is a number of peaks is a very large. Many numbers of peaks are coming in this region due to the winding of the molecules or the atoms. So this is used a fingerprint region. And the identification of the group, which of the material is present, whether this contains the CH2 group, CH3 group, or OH group, like this. So this is a functional group region, 4000 to like this, 1500, 4000 to 1500 centimeter inverse. And this further can be divided in the four region, whole spectra. This fingerprint region itself one, and this function group divided in the three region: C double bond, triple bond. In double bond, CC, CO, or CN. These functional group. And this is a triple bond, CC and CN. And this is a CH, OH, or NH. So this should be mind. And further, the peak position of any bond may be slightly vary, which depend on which molecules is attached, or the which is neighboring molecules, neighboring structure. So further, as we saw in the last slide, this can be further understand in better way. This is a fingerprint region. There are many peaks is coming. This is called the fingerprint region, CO, CN, or CC. This is a double bond region, CC, CO, or CN. Triple bond region, and this is a OH, NH, and CH stretching. So this can be understand better way by this spectra also. Further, this can be further understand or better way And the triple bond 2000 to 2500 centimeter in reverse. And this is the alkenes. C triple bond C are typically weak and sharp, around 2100 to 2260. CC bond, C triple bond C, and nitriles C triple bond N are the strong, sharp, peak in the same region, often the higher end. So this is the same region, but this is strong or this is typically weak. In the double bond, carbonyl group C double bond O is a very weak sharp, very weak, very strong, sharp peak around 1710. This is very important in many compounds. A single peaks will be very helpful for identification of a particular compound. For the ketone and the aldehyde, it can be shifted slightly for the other carboxyl like ester or the carboxyl acids. Alkenes C by C appear as a big peak and this and this. So means this is a peaks 1710 centimeter inverse to the CO, but this can be very slightly depend which type of the molecules or the material or which type of the items are nearby this CO and C group. So, and another is a hydrogen related stretching, as we saw in the previous slide, 2000 to 3600 centimeter inverse. Then this OH come in category is very broad and strong absorption, 3200 to 3600 centimeters inverse for alcohol, or a strong broad band from 2500 to 3300 centimeter inverse for the carboxyl acid. So keep in mind which type of the material we are going to analyze. And this is NH group, this come 3300 to 3500 centimeter inverse, and CH 3000 centimeter inverse for the sp3 hybridization, and CH this for the sp2 hybridization. So these are the details. Whenever we are going to compare the materials or identification the material, these peaks are very important. So keep in mind. Thank you, my dear friends. For any further details, you can contact to me at this address. If you think this is a slightly valuable, please like, comment or subscribe my channel. Thank you.